you your revision sets. I know that some of you may have done some of them already, so it's fine. For those of you who haven't, I hope this would be helpful for you. For those of you who have done, at least this will become just a reminder, or just a quick revisions for those of you who have done it. Anyway, so we're going to go through question by questions. For the first question that I have here, okay, so this is basically asking you to complete the table using a tick and a cross. Now, please remember that when it comes to exam, you must follow the instructions. If they ask you to put a tick and a cross, do that accordingly, just like how they give you an example here. Now, to show that the cell structure can be present in a particular type of cell, and if it cannot be present, so put a tick or cross in every box. So you have to put that for all of the box, and the first one has been completed for you. Now, this one we have a box of eukaryotic and prokaryotic cells. So what are the informations that we need to take note of? So this goes back to the definition, whether you remember what is the definition of a eukaryotic or / prokaryotic cells or organism. Now I want you to remember, okay, eukaryote. Okay, so eukaryotic cells, okay, they're defined by any cells that has a, remember, membrane, okay, bound nucleus, and then membrane bound organelles. So please take note, alright? So why do we say membrane bound? Okay, because we know that these organelles have their own membranes, okay? So nucleus, because what we learn is in prokaryotic cells, the one that we have to learn for our syllabus is we look at the structures of bacteria, okay? Specifically, right? So bacteria, okay, they do not have a membrane bound nucleus, right? Okay, instead it might have, okay, their what do you call this, circular DNA or plasmids lying around within its cytoplasm. It's not in like a proper nucleus that we are aware of. It does not have membrane bound organelles, but it does have something else, at least one organelle that we can actually name, so which is what we need to go through and proceed with answering the questions. All right, so okay, so the first row has been ticked for you and crossed for you. We know that eukaryotic cells have a membrane-bound nucleus. Okay, prokaryotic cells does not have. In the case of bacteria, it does not have that, but instead it might have what you can take note of. Okay, it might have instead, okay, circular DNA and plasmids. Okay. In case you don't know by now, please remember that plasmids is what we learn in our syllabus as a role, for example, in antibiotic resistance. So you must recognize and know what plasmids are already. Golgi body is an example of a membrane-bound nucleus. Membrane-bound organelles. So eukaryotic cells, it will have Golgi body. Okay, prokaryotic cells will not have a Golgi body because this is a membrane-bound organelles. Circular DNA, eukaryotic cells and prokaryotic cells. So do eukaryotes have circular DNA? So do eukaryotic cell have circular DNA? Yes, it does. And prokaryotes, yes, it does, because I've just mentioned that earlier. Here's circular DNA. Now, where exactly in eukaryotic cell can we find the circular DNA? So that can be found in the mitochondria and the chloroplast. Alright, and the next one, last but not least, ribosomes, 70S ribosomes. Okay, that's the smaller ribosomes. Okay, it can be found in prokaryotic cell. Yes, that's one of the features that you need to remember for bacterial cells, but don't forget that eukaryotic cells will also have it as well. Now, where can we find them? Again, it is in the mitochondria and chloroplast. Okay, this is a quick reminder that mitochondria and chloroplast, they were actually evolved from bacteria. So that's why they share the same features that a bacterial cell might have. Okay. Moving on with the next question, all cells have a cell surface membrane. This is showing a transmission electron micrograph of two adjacent animal cells. So you have cell 1 here and cell 2 here, side by side with one another. Alright, and then they wanted us to draw a diagram, okay, to show the 4 dark lines, and we must label the diagram to identify what the dark lines and each of the spaces between them are showing, right? Just like any biological drawing, make sure, okay, your drawing is clear, clean, and continuous line. So let's have a look at the drawing that I have done. Okay, now they specifically mentioned that we must draw in the region that they have provided here, which is in this box. Okay, so that's exactly what I've done. I only drew the one that is in that box. All right. Okay, and the rules of microscopic drawing is also applied. It should be applied on this as well. Okay, remember when you're drawing, right, you must always draw in clear, continuous line. Now, I know on my presentation here, on my video here, I'm using a pen because I'm writing this on a tablet. If you are writing this on your answer paper, please use pencil. Okay? And make sure that you can erase your diagrams properly. Erase cleanly, right? Just like what I have emphasized, what I've reminded you in terms of biological drawing in your practical, it should also apply here as well. Right, so because it's asking you to label, right? So I have done here, so the 3 marks, okay, the points that they want to see, okay? We must show 4 dark lines. I have 4 dark lines, 1, 2, 3, and 4, which is again representative of what we can see here. Where does the 4 dark line comes from? Okay, that's 1, 2, 3, and 4. And now we are going to label. So what are those 4 dark lines representing? Okay, right? So if you look here, what I've done is, this is the hydrophilic phosphate head, because you must recognize that this is the cell surface membrane. So these 2 structures are your cell surface membrane. Okay. So by the way, these are your notes. This is not what they want you to label. Okay, my label is the one that is in blue. So these are your cell surface membrane, which means that the dark lines here that we can see is representing the hydrophilic phosphate head. All right, and the one here at the center that you can see here, which I have again labeled with an arrow, with a line, ruled line with a ruler, right? That is where our hydrophobic fatty acid tail, or also core, is, right? Hydrophobic core. So please remember that. And then the next point that they want you to be able to recognize and realize is that the space between these 2 membranes, because this is between 2 cells adjacent to one another, so this is the space between 2 cells, or intermembrane space. Intermembrane. I don't really like to use the word intermembrane because I'm scared that you guys might get confused, but instead you can just label the space between 2 cells. That's clear, simple, and then I think that is also acceptable. All right, so please take note, and that's how it is. Okay.